Hi, I'm Ruth Fishman from Koren Wildlife Hormones and Behavioral Ecology Group in Bar-Ilan University. Females may choose different mating strategies. Among them is multiple paternity. Multiple paternity, the phenomena whereby many fathers contribute to an individual litter, is common throughout the animal kingdom, from insects to mammals. With the use of genetic methods, it was found in more than 100 mammalian species, even those that were considered monogamous. In males, the benefit of multiple mating is obvious. Females, however, are usually limited by the number of overproduced or offspring raised, making the benefits of multiple male partners less obvious, while there are many costs. The possible benefits to females can be direct and indirect. Direct benefits can be paternity uncertainty to counter infanticide by males, avoiding sexual harassment, gaining material benefits, as we can see here, for example, the cardinal male feeding the female, parental care provided by the male, and also fertility assurance and increasing litter size. Indirect benefits can be acquisition of good genes if the partner has a lower genetic quality compared to another male. Mating with the additional male may give the female's offspring a genetic benefit. For example, genes that make the offspring more attractive to the opposite sex and enhance their reproductive success, or genes that enhance offspring survival. Another possible benefit is increasing offspring genetic diversity. This might allow a differential survivability of the offspring under different conditions. While in the first option selection might slowly select the same genes and decrease the genetic diversity, the second option works in the opposite direction, preserving genetic diversity across generations. In this study, we focus mainly on the genetic benefits that multiple paternity might provide the nutria. The nutria is a semi-aquatic rodent native to South America. Currently, it's a successful invasive species worldwide, considered to be one of the worst invasive species. It breeds year-round, producing large litters. We previously found that females in better condition and having lower testosterone levels have male-biased litters, suggesting a certain cost of testosterone to female fitness. Another interesting feature of this species is its tendency for selective abortions. When in good condition, females been observed to abort litters composed of females and immediately after conceive litters composed predominantly of males. The nutria has a polygamous mating system, and its group structure comprises juveniles, several adult and subadult males and females, and one dominant male. The dominant male is territorial, differentially larger than the other adult males, and initiates almost all of the aggressive interactions in the group while receiving none. We wished to examine whether multiple paternity occur in the nutria and what benefits it may provide. In Agamon-Hula Lake, like other areas worldwide, efforts are made to regulate the overpopulation of this invasive species. We collected carcasses from culling efforts and performed post-mortem dissections. Since the nutria fetuses start to grow hair at the last trimester of its 4 months gestation, we used fetal and maternal hair to quantify steroids. The steroids accumulate in the hair, thus providing a long-term measurement of their levels. We also took tissues from the mother and fetuses for genetic analysis. We tested for 7 microsatellites, which are short repeating sequences that show high polymorphism between individuals. For every fetus and mother, we determined the length of the alleles for every microsatellite. And using colony software, performed simplicity analysis, which related every fetus in the litter to a virtual father based on its allele combination and the alleles of the mother and the rest of the litter. As you can see here, in this litter, all the different combinations of alleles could be inherited from a single father, while in this litter, the allele combinations could be only explained by 3 different fathers. We found that only in 41% of the litters there was a single father, while the rest of the 59% had multiple fathers. The number of fathers in a litter ranged between 1 and 4 fathers. We categorize the fathers as single father, related to all the fetuses in the litter, main father, related to most of the fetuses in the litter, and rare father, related only to 1 or 2 fetuses in the litter. These 3 groups were statistically different.
First, we wanted to know if there is any difference between females impregnated by multiple males and females that are not. Higher weight, of course corrected for litter size and gestation age, can indicate condition, and length measurement can be indicative of age. However, there was no difference in these parameters between females having litters of multiple paternity and single paternity. The females' cortisol levels could also provide some clues regarding their condition. We examined that while correcting cortisol levels for gestation age, because in the last stages of gestation, maternal cortisol is significantly higher in the nutria like in many other species. Again, we did not see any difference. As we know from prior research, nutria females having lower testosterone levels have male biased litters. However, the females' testosterone levels didn't relate to whether they were impregnated by one male or multiple males. So overall, it seems that being impregnated by one or multiple males is not related to any special attributes of the nutria female. In order to examine if multiple paternity benefits the nutria by producing fetuses with better morphological features, we examine if the fetuses related to the rare father differ from those of man or single father. We checked various morphological features, such as weight, total length, hindfoot length, and more. However, we didn't see any difference. In polygamous species, having male offspring can increase the female fitness. However, also regarding the sex of the fetuses, there was no difference between fetuses related to the different fathers categories.
However, when we looked at the testosterone levels of the fetuses in relation to the father's proportion in the litter, that is, how many fetuses are related to the father divided by the litter size, we found that male fetuses' testosterone levels are positively related to the father's proportion in the litter. For females, however, there was no association. When we looked at the male fetuses' testosterone levels in relation to their fathers being single, main, or rare, there was no difference between fetuses relating to single and main fathers, but fetuses related to both of these fathers categories had significantly higher testosterone levels than fetuses related to the rare father. These are very interesting results, since in the nutria group the dominant male, which is observed to have a higher reproductive success, is territorial and differentially aggressive, traits that are linked to higher testosterone levels. The dominant male is also differentially larger, so we examined whether there is an association between adult nutria males and their testosterone level. We've seen like in the fetuses, adult males have higher testosterone levels compared to females. And we found that in adult males, bigger males have higher testosterone levels. As you can see here, we measured weight, total length, and also skull length, which is sexually dimorphic feature linked to testosterone levels in some species. We measured also additional morphological features, all showing this positive correlation. In adult nutria female, however, there was no relation between body size and testosterone levels.
Some of the litters contained fetuses that were much smaller than their siblings, as you can see here, or even shriveled. We defined growth retarded fetuses as weighing no more than 80% of the litter's mean weight and being lower than the 25th percentile. These fetuses have a lower chance to reach full term and would probably be resorbed or aborted. Interestingly, growth retarded fetuses are mostly males. and they are significantly more common in the early stages of pregnancy. As the nutria is known for selective abortion and fetal resorption, we wish to know to which of the father's categories these growth retarded fetuses belong. As we can see here, the growth retarded fetuses belong almost exclusively to single and main fathers. But since the frequencies of paternity of these fathers are much higher, we cannot conclude with this small sample of growth retarded fetuses that we are observing a mechanism of potential resorption of fetuses related to main and single fathers. However, when we look at these growth retarded fetuses in the multiple paternity litters and examine what would happen if they would be culled along the pregnancy, we see that the litter size is, of course, being reduced when these fetuses are being culled. But the number of fathers does not change. Even in this 1 litter, where a fetus was related to a rare father, it was one of the relatively few cases in which the rare father seed 2 fetuses in the litter. So the number of fathers didn't change. Reducing the litter size while keeping the number of fathers leads to a higher proportion of fathers per litter size. And this difference between the proportion is significant. So eventually the number of fathers is not reduced, meaning that the nutria does not use fetal resorption for reducing the number of fathers. We also see that the number of fathers is not reduced with the progress of gestation. And interestingly, there is also no association between litter size and the number of fathers. So in the nutria, multiple paternity doesn't provide a benefit of increasing the litter size. In conclusion, we did not see any morphological advantage to mating with a rare father. We've seen that male fetuses related to rare father have lower testosterone levels. We do not know whether that predicts the adult testosterone levels, but we do know from studies of intrauterine effects and prenatal testosterone administration that fetal testosterone affects features associated with reproductive success. And in male fetuses, higher testosterone levels are related to higher attractivity, aggressiveness, and reproductive success in adulthood. So, in that respect, mating with the additional male would not benefit the female with more attractive male offspring. We've seen that the number of fathers is not dependent upon litter size and also does not decline with fetal resorption. So, the number of fathers is being conserved. Having offspring related to multiple fathers creates genetic diversity. This promotes inbreeding avoidance and increases the offspring genetic diversity. Genetic diversity is particularly adaptive for invasive species as they face the challenges of adaptation to new environments and a decrease of genetic diversity due to the founder effect and genetic bottlenecks. In conclusion, in the nutria, it seems that the females favor genetic diversity even at the possible cost of reducing the future reproductive success of some of their sons. I would like to thank Dr. Dvorah Matas, our lab manager, the members of the Koren lab and Vortman lab, our undergraduate student assistants and volunteer, and Kakal JNF for founding the Nutria project and supporting the Hula Research Center. Thank you very much for listening.